I am Professor Krishnan from Department of Biochemistry, Central University of Rajasthan. Now I am going to give the talk on metagenomics, a novel approach for bioremediation. Bioremediation, it's a very important topic nowadays. Bioremediation is a waste management technique that involves the use of organisms to remove that neutralize the pollutants from the contaminated site. According to the IPA, the bioremediation is the treatment that uses naturally occurring organisms to break down that hazardous substances into the less toxic or non-toxic substances. Advantages of bioremediation. Bioremediation may result in the complete metabolism of pollutants, are considered to be highly effective and environmentally friendly strategies. So far, the bioremediation was done by various processes by using the fungi, plants, bacteria. By that, the pollutants can be removed from the environment and then environment reach a unpolluted environment. By that, we can bring the earth as a green earth. Now, the microbial bioremediation is taken as a part. Why? Because aerobic and anaerobic catabolic strategies, microorganisms, to degrade the huge range of organic compounds present in the ecosystem. Because pollutant molecules are often structurally similar to the natural molecules, there are always some microorganisms in the contaminated ecosystem that are able to metabolize pollutants which serve as their main carbon source. During the microbial degradation, all changes in the chemical structure of pollutants are due to the action of enzymes whose specificity is often broad enough to accommodate several molecule similar structures. Once identified and isolated, these enzymes can therefore be engineered by directed evolution to improve their stability or efficiency with respect to the particular compound. Then what is metagenomics? After talking about that bioremediation, now I want to introduce the topic metagenomics. First, before I am getting into the metagenomics, what is the metagenome? Metagenome refers to that idea that collection of genes sequenced from the environment could be analyzed in way analogous to study a simple genome. Metagenomics, either you can call it as environmental genomics, ecogenomics, or community genomics, is the study of genetic material recovered directly from that environmental samples. The metagenomics application is a modern genome technique to the study of communities of microbial organisms directly in their natural environment by bypassing the need for isolation and the lab cultivation of the individual microorganism or species. How you can sequence it? The ones that materials or DNA isolated from that environment directly by uncultured way, you can go for the sequence by using that modern technology called next generation sequencing. The next generation sequencing, otherwise called NGS, also known as high throughput sequencing, is that catch all term used to describe a number of different modern sequencing technologies, including Illumina sequencing or Roche 454 sequencing. What do you mean functional metagenomics? The functional metagenomics, which consists in assigning the functions to proteins encoded by all genomes of a microbial community with no isolation and cultivation step. A highly efficient way to boost the discovery of novel by a catalyst from the huge diversity of uncultured microbes. Screening for pollutants degrading enzymes, that is a two complementary approach, can be used to identify pollutant degrading catalyst in microbial communities. Gene sequencing analysis and functional annotations based on the content of available sequence databases. The other is guided by the observation of pollutants degrading phenotypes harbored by recombinant metagenomic clones. Here I am giving the total outlook of that methodology. In this slide, you can very well see that the microorganism collected from that environment and then the genetic material will be isolated. The isolated genetic material will be restricted by the restriction enzyme and then it can be cloned in a known plasmid. After cloning the plasmid, it is called a recombinant plasmid, can put it into the host. Now it is called genomic library. The genomic library can be taken in two ways. Number one, you can put it in that one side to express this DNA or genetic material and then you can characterize it, the efficiency, and it can go for the application. Otherwise, the gene sequence can be go for to diversity analysis and find out the novel sequence which already reported. Now, the next slide is going to give that flow diagram of what I described the earlier slide. So, now you know that how a DNA genetic material can be isolated and then how it can go for that. One way it is going for the metagenomic analysis, another way you can go for cultivation method. But finally, what we are going to do that how a enzyme, a novel enzyme can be isolated from the sample and it is going to be used for the environment safety. Now, the activity based metagenomics for the discovery of new tools for bioremediation. So, the technology relies on that observation of phenotypes linked to the reactions involved in breakdown of the targeted pollutants. It has three prerequisites: the cloned DNA or cDNA fragment between 2 to 200 kbb in size length into the expression vector, either plasmid or cosmid or even bacterial artificial chromosome for the creation of metagenomic or metagenomic libraries respectively. The heterologous expression of cloned genes into the microbial host, design of sensitive phenotype screen to isolate the clones of interest with the targeted activity, also called the screening hits. Now, this is the whole scenario what I described for the last 10 minutes. That environmental sample has been isolated, the DNA is isolated from the environmental sample and then it is going to be restricted, cloned in a proper vector and host, then you are going to screening. One side you are going to look into the enzymes, another side you are going to look into that what you called the biodiversity. Right, this is a classic example I am taking to explain very clearly what I narrated for last few slides, that is environmental pollutants. Now, this slide shows that you no need to tell about it, where we have the lot of pollution in different areas through various purpose. Now, if you are going to the next slide, how that the annual number of premature death is going on from large folding increase. So, this is the time we have to take the novel technology, not only control the pesticide, at least we have to manage the pesticide in the environment. The next slide is going to tell about that how many deaths occur in each category, it is explained very clearly. Now, the bioremediation of pollutants through metagenomics with a case study I am going to explain you. Now, the cultivation based methods elucidating only small fraction of the total bacterial population, in particular rare bacterial dexa, were undetectable and resulting into that underestimation of the population diversity actually present in the natural environment. To overcome these limitations, culture independent molecular techniques have been developed to study the total microbial diversity. The understanding the microbial diversity and its function in complex environment has been improved by next generation sequence. This slide clearly indicated that even by literature you can see that only 1% of that world microbial population is culturable and studied very well. But this is the new technology, that's a metagenomics, where again I repeat it, the genetic material you can collect it from any environment and then without any culture you can go for that this type of studies. The next slide is going to explain what is the case study. Here it's very important for that what you call the beginners of metagenomics, where we have collected the sample from a site where you have the ternary effluent treatment. From there we have collected and samples divided into two groups. One group culture dependent, another group culture independent approach. When you are going to see the further slides, how that culture dependent approach is vary from culture independent approach. That huge variation you can study or you can see when coming slides. Right, this slide it's definitely can explain that you have that strategy, the diagram representing of that percentage of bacterial community in RE, that is you can have that pollutants collected from different area, that is raw effluent means that RE, then primary treatment is called PT, ST is a secondary treatment and TT is called tertiary treatment from the GTP, that is called where that the treatment plant is going on in ternary side. Now you can see that number of the microorganisms available either RE, PT, ST and DT is very well explained by pie diagram. But if you are coming to that high throughput 16S RNA sequencing unexplored by CETP sample, now you can see that even 2 base pair 2, 100,000 base pair you can sequence it, and then you can go for that the refraction analysis representing that species and reads into different stages of CETP. Now this different the graph of the lining express that the microbes level with a different treatment plan, as I told that where the raw effluent, primary treatment, secondary treatment and tertiary treatment. This graph very well explained that how that available of the microorganisms or genetic material vary from one treatment to another. Right. So this is that family level phylogenetic 3 different effluent sample by CETP. Now it's very well explained. Huge number of that microorganism can be identified by using this phylogenetic tree by collecting the sample from the directly from the environment. It's not cultured, it's culture independent method. Right, this is that a recent approach, heat map analysis showing that abundance of bacterial class in effluent sample of CETP. Now you can see that the first column is going to apply the raw effluent, then second one is primary treatment, secondary treatment and also the fourth one is primary treatment. Now if you can compare that, why you need this type of analysis, you can see that the green color, specifically you can take the green color. The green color specifically showing the only one treatment plan where that microorganism can be exploited further. Then we have the Venn diagram showing the unique and concomitant species among the different effluent samples. Now you can see that we are comparing that raw effluent, again primary treatment, secondary treatment and tertiary treatment. So how many samples or how many microorganisms is available in each sources, you can identify that. But this graph show that what are the microorganisms available, the RE to PT, PT to ST, ST to TT or DT to RE. Such a way you can see that, or other way you can see that what are the microorganisms unique in the treatment plan, either in the raw effluent or primary treatment, secondary treatment or tertiary treatment. Now, comparatively you can see species obtained from the conventional and NGS approach. NGS approach, I told that already, next generation sequencing approach. This is not culturable, culture independent method. So directly we are taking that genetic material and sequenced by the NGS. Now it's this graph, you can understand that what I try to tell about that, what you called the 20 minutes of my talk. Number one, RE, that is I told that raw effluent, where by culture dependent we can say 15 species only, but at the same time if you are going for metagenomic, you can see 605. I don't want to explain. If you are coming to the total, only 45 we can go for the culture method, but if you are coming to that culture independent method, if you have the 2156 species. So this is the way you can collect lot of genetic material which is not possible to culture in the laboratory can be analyzed. So the exploration of the novel genes from the metagenomic library. Now you can see that, now as I told earlier, described it, how I am going to prepare the metagenomic library. So we are collecting the sample directly from the environment and then we are preparing the metagenomic library where you can, this slide very well know that how you can, the genomic libraries looks like in your agarose gel. Then that gene can be isolated and then put it in the vector. As I told that the vector we have taken is PUC 118. So this is that not for expression vector, this is a cloned vector where you can increase the copy number that can be analyzed further. Now by having this sequence, or we have that DNA cloned up, collected from the genomic library, compared, and we have isolated the DNA library or gene isolated from that environment can be compared with which is available already in that genomic library. By that you can identify the novel genes which are present in the polluted area. This further exploited to go for that what you call the application. But if you are coming to the next one, why we have selected that ternary effluent treatment plan? Here it's very well understanding that this genetic material, or we are looking for the novel tannase enzyme which can degrade the tannic acid. Why? Because the tannic acid is stable in the environment, it's giving lot of problem in the environment or the public. So we want to isolate the novel tannase enzyme from that uncultured method or culture independent method. By that can applicable very well. By that you can remove the tannic acid from the environment. Here I want to register some information because we are doing this work. There is a place in Vaniyambadi, Tamil Nadu where the tannery industry is a huge number. So they need a strong technology to remove that tannic acid in the environment. By that we can bring that tannic or that tannary effluent as a useful water for the public. So, by that we are screening that tannase enzyme from or collected or expressed from the DNA collected from the environment, and then we isolated this tannase enzyme gene and we characterize it. Now we identify the novel enzyme. Now its time is not very far. We can give the novel tannase enzyme to reduce the tannic acid in the environment. So through this one, we can go for that tannic acid free environment in the tannery industry side. Other than that, why we need the metagenomics? Discovery of novel natural product from the environment, novel antibiotics, new molecules with the new functions, new enzymes and bioactive molecules. What is the genome or species? Diversity of life, interplay between the human and microbes. How do microbial communities work and how stable are they? The holistic view on biology you can establish by learning more about the metagenomic and their application in various fields. By that, I can give the short information about that a novel techniques, a newly developed techniques where it can apply in the different field as I told that. So anybody can learn it. Now earlier doing metagenomic it's not possible because it's highly money oriented or you need lot of grant to work on that. Now since so many laboratory now started the metagenomics, now you can go for outsource of sequencing your DNA by NGS method and you can apply the field where you are working. By that I can say we all together work on this new field, we can make our world a better place to live without any pollution. At least we have to take the oath that we have to bring that or we can manage the pollutants in the environment. Thank you very much.